Good morning, juniors. Today we are going to talk more about what goes on in cells, specifically about two processes called osmosis and diffusion. Our objective for today is I can explain the process of osmosis and diffusion in cells. A quick review from yesterday. This blue structure outlining the cell is labeled as a plasma membrane. That's another word for cell membrane. And the cell membrane has a really important function for all cells, plant and animal cells. The cell membrane, or the plasma membrane, acts as the guard of the cell. It controls what enters and exits the cell. So it can let water in and out of the cell, but it won't let other important molecules like maybe a certain type of protein out because the cell needs them. Let's get started with our notes for today. Cells need things that they don't have immediately available to them, like water, like oxygen that we breathe in, like glucose that comes from our food. The cell needs to be able to get these things and in some cases get rid of certain things like waste. In order to do this, cells have a cell membrane, which you guys are already pretty familiar with. But we have a special science term to describe cell membranes. We say that they are selectively permeable. This means that only some things can move in and out of the cell. For instance, water may be able to move in and out of a cell, but DNA may not. Sometimes we say that the cell membrane is like a screen because certain smaller things like air and water can get through the little holes, but bigger things like a soccer ball or maybe a person's hand can't. Your next key point is this new term called concentration that you may have learned about last year with Miss Anselmo. Concentration is referring to how much stuff or how much matter is in a specific space. So right here in this box that I'm outlining with my mouse, there's a high concentration of molecules. There's a lot of molecules all tightly packed together in one space. So this is a high concentration. In the box to the right, there are fewer molecules and they've got a lot more space in between them. None of them are touching. They're not really within very close distance to any other molecules compared to this box. So we say that this is a low concentration, less stuff in the same amount of space. This term up here on the right, equilibrium, you don't need to write that down, but that just means equal. Equilibrium, that EQU, that prefix, it's in both words, means the same concentration. So diffusion, the first process that we're going to talk about, is the process of molecules moving from a high concentration to a low concentration. So in this diagram below, the molecules start out really close together like we just saw in the high concentration box. They're touching, there's not really any space between them, but they're going to diffuse out from a high concentration to a low concentration. So in this middle picture, they've got a little bit more space between them. They're moving away from where they started. And then in this far right box, there's a lot more space between them than when they started. And they're basically filling up the whole spot. So they've diffused from a high concentration over here, closely packed together, to a low concentration over here. In the video that I'm about to show you, you will get to see food dye, specifically red food coloring, diffuse through a sample of water. So here we have the video. It's just a sample of clear water, and you're going to get to see the red food dye diffuse through this sample. So you see the drops just went in, and they're already beginning to diffuse throughout the sample of water. They're spreading out. The red color is becoming less intense because those dye molecules are moving out. They're getting a lot more space between each other. The concentration is decreasing. The molecules are becoming less tightly packed together. They're moving from a high concentration to a low concentration. They're diffusing. Awesome. Here's just a snapshot of the process that we just watched in that video. You see the dye starts out really dark, almost black right here. And as it diffuses through the sample of water, the color becomes less intense. Those dye molecules become less spread out. More spread out, more space between them. They've gone from a high concentration, tightly packed over here, spread out, a low concentration here. Here's just another diagram showing that molecules move from a high to a low concentration during diffusion. Our next term is osmosis, that second process we said we were going to learn about from our objective. Osmosis refers to specifically and only the movement of water through a semi-permeable membrane. We learned what that word semi-permeable meant already. Semi-permeable membrane represented by this dotted line right here, and these shapes right here, these are water molecules. That's an oxygen and two hydrogens, H2O. So if we start on the left here, we've got 1, 2, 3, 4, 5, 6, 7, 8 water molecules on the left, 2 on the right. The water molecules can pass through the semi-permeable membrane, so after some time has passed, the water molecules, through osmosis, have moved through the semi-permeable membrane, and now we have equilibrium. We have 5 water molecules on each side of the semi-permeable membrane. We're at equilibrium. In this next video, you're going to get to see in really, really sped up fashion how osmosis works in real life. Now I know the music is a little bit silly, but bear with me. So this is what the lettuce looked like before. The lettuce looks kind of wilted. So it's super sped up, 720 times faster than real time. We can see the lettuce is starting to expand. Those cells in there are taking in lots of water. It doesn't look so wilted anymore. It looks stronger. That looks way better than the lettuce that we started out with, because water has moved from osmosis. There we go. Water is moved from via osmosis back into the plant cells. So, in osmosis specifically, when water is moving, there are 3 types of solutions or mixtures that we can classify. You don't need to write this down. I just want you to become familiar with these terms. I'm going to use them and you're going to hear them in classwork, but you don't need to put them in your notebook. The first type of solution is a hypotonic solution. That's when there's a high solute or high molecule concentration inside the cell. The cell actually gets bigger because water enters the cell to spread out all of those molecules. Those molecules don't want to be close together. They don't like that. So the water enters the cell gets bigger. That's what's happening right here. water goes in, cell expands. The second type of solution is isotonic. That's when there are equal concentrations of molecules inside and outside of the cell. There's a balanced flow of water in and out. So water is still moving in an isotonic solution. There's just no net gain or loss for the cell. Let's see, we have water in, water out. Everything's normal. The cell looks normal. The last type of solution we can have is a hypertonic solution. That's when there's a low concentration of solute outside of the cell. A high concentration of solute outside of the cell. The slide is wrong. The cell, like you can see right here, shrivels up because water is leaving the cell. The water wants to spread out all of those molecules outside of the cell. So water leaves the cell loses mass, loses water, and shrivels up like what's happening right here. Here's just another clearer picture of hypotonic, hypertonic, and isotonic solutions. In this hypotonic solution, actually I like to think of hypotonic like hippo. Hippos are really big, so in a hypotonic solution, the cell gets bigger. In this hypertonic solution, all the water is leaving the cell because look at all those molecules on the outside. Water's leaving the cell, wants to spread all those outside molecules out. The cell shrinks. Isotonic solution, we've got equal water in and out of the cell. No net loss or gain of water. The cell stays the same.